Dear students, my name is Tomáš Rendek, and along with my colleagues Daniela Klimová and Jakub Styk, and under supervision of Professor Repiská, we as PhD students of the Institute of Molecular Biology, Genetics and Clinical Genetics of Medical Faculty of Comenius University prepared for you this lecture about detection of mutations in clinical practice. We selected for you 3 topics which we believe are very interesting, and they are also in direct link with your lectures. And we will cover 3 main issues, which are mutations with gain loss of function, prenatal diagnostics, and personalized medicine. Those 3 topics will be discussed mainly superficially, because those are all large topics, but nonetheless, we believe it would be very helpful for you, and we can of course discuss these problematics later on in other years, as well as privately via consultations. So we can start right ahead with gain of function mutations. The gain of function mutations are types of mutations in which the altered gene product possesses basically new molecular function or a new pattern of gene expression. It means that gain of function mutations are almost always dominant. Of course, they can be also semi-dominant, but it's not really important right now. So we can summarize those are dominant mutations. They encode a new abnormal function of proteins. They accelerate tumor growth, which is very important, of course, in clinical practice. And the same specific mutation is in all affected individuals. And we can see here the very first example, which is achondroplasia. Achondroplasia is autosomal dominant genetic condition, which is very famous, probably the most famous cause of dwarfism. We can see it, for example, from Lannister in Game of Thrones. And it is basically a point mutation at position 1136 in chromosome 4. It's a transition or transversion. And basically the gene that is targeted is FGFR3, and basically this mutation increases its activity, which significantly restricts bone growth. So the person is logically small in growth. Another very interesting example is Huntington chorea. Huntington chorea is basically autosomal dominant disease, and it is very famous. For example, if those who watched Dr. House, Dr. 13, her mother had it, and of course she inherited it. Since it's dominant, there was 50% chance she would get it. And this is basically characterized by expansion of CAG triplet. For those who remember from biochemistry, CAG is glutamine amino acid. So basically, this is also called polyglutamine disease. And this excessive glutamine is basically harming the neurons, and the more repeats are genetically inherited, the earlier this is manifest. So maybe, for example, in the movie or in the Dr. House, her mother had it around 40 or 50, but number 13, she had it early in the 30s, because she basically it is a phenomenon called precipitation, where the number of expanded triplets is greater with each generation passed. Now we can switch on the loss of function mutations. Loss of function mutations, also called inactivating mutations, result in basically in a gene product having less or no function, so being partially or wholly inactivated. When the allele has a complete loss of function, which means also it is a null allele, it is often called also amorph, but this is not a term that is widely used in clinical practice. So what we must remember is that those are recessive gene mutations. Usually they are targeting an enzyme. and basically mostly they are deletions, stop mutations, or frameshift mutations. Another very good example, which is also widely used in education of young geneticists, is alpha thalassemia, which is basically a deletion of HbA1 or HbA2 genes, which are genes responsible for formation of alpha globulin, which is a subunit of hemoglobin. And we can see here on the picture even the severity of the disease as it promotes with adding more and more mutations, as we see on the left, a normal phenotype and genotype, and then a carrier, alpha thalassemia minor, which is basically also asymptomatic, and then with basically 2 and more mutations, we have a fully developed alpha thalassemia, and with missing all 4 copies of HbA gene, basically we have a lethal disease. Another 2 famous, or rather infamous, examples are Duchenne muscular dystrophy and spinal muscular atrophy. When we look at the Duchenne muscular dystrophy, it is basically an X-linked recessive disease, which also means it primarily affects boys because they need to have just 1 damaged copy to fully develop a disease. Here also it is important to see the length of the actual gene. Dystrophin gene is one of the longest genes in the human genome. It has 2.6 million bases, but only with 97 exons. And of course, it's not the longest. The longest is titin, which is also expressed in muscle tissue. But having such long genes have its risks, because within this very long gene, when there is only 1 mutation that causes a shift of the reading frame, it means the entire protein would be translated differently, and we have really a severe Duchenne disease. And the next one, the spinal muscular atrophy, is basically a rare neuromuscular disorder, and it results in the loss of motor neurons, and basically it causes a progressive muscle wasting. Here in the picture we have the most common form, which affects basically a defected survival motor genes, but SMN genes, but we have to remember that this is just simply a group of diseases, and some of them are even X chromosome linked. And for this type of spinal muscular atrophy, there is also available a gene therapy which got recently very famous. It's called Zolgensma, and basically it was approved for children less than 2 years old in 2019. It has been used also in Slovakia for some children, though it's very expensive. And of course, we cannot omit the most famous and very well studied genetic disease which causes inefficient blood clotting. It's hemophilia A. There is very good to mention that the genetic heritability is only about 70%, because it is X-linked, of course, as you may remember even from your previous studies. But there is one interesting phenomenon, and those 30% remaining, which are not inherited from your parents, are de novo mutations. They are mostly caused by insertion of retrotransposon LINE1 into the F8 gene. Basically, this is a movable genetic element which is predisposed to insert into exon number 14 of F8 gene, and it causes basically de novo hemophilia. Cystic fibrosis is a genetic disorder affecting mostly lungs, but also pancreas and intestine, although it causes almost always a male infertility. And this is also autosomal recessive disorder, which we know mostly by the most famous mutation F508 deletion, which is basically deletion of phenylalanine in the CFTR gene. We have to remember though that this is not the only single mutation. It will be not that easy. It is until now identified more than 900 mutations causing cystic fibrosis to a different expression of the actual disease. And it's very interesting because this disease is relatively common. It is 1 in 3,000 people are diagnosed with cystic fibrosis, and it is believed that it was spread throughout the old continent and then throughout the world. Of course, because the heterozygotes have an actual advantage, and it is believed to help people survive diseases like cholera or difficult diarrheas. It is also very good to mention that there is a rule which can be applied to gain of function and loss of function mutations. It can be applied though very loosely, and it is that mutation in the non-coding regions, such as promoters, introns, and untranslated regions, are usually associated with loss of function, and mutation in coding region, mostly those are point mutations. They are associated with gain of function, so abnormal interactions or incorrect aggregation of proteins. Now we would quickly switch to another of the three main topics, and it is prenatal testing. Fetal abnormalities cause up to 20% of perinatal deaths. And advances in the prenatal genetic testing, which is in the past, let's say, 20 years, offer a great opportunity for identifying high-risk pregnancies. In current clinical practice, various options of prenatal testing are available. Prenatal testing has passed a really long way since those traditional invasive methods, such as was, for example, amniocentesis, choriowheel sampling, or chordosynthesis, which are all very invasive and also very dangerous and uncomfortable for mothers, towards really a non-invasive procedure based on blood sampling. In this picture, for example, you can see the timeline of prenatal testing of Down syndrome, with the very first diagnostics happened in 1986, which was really invasive, towards 2010, when we already have available basically prenatal testing based on blood sampling. When we look just quickly into the history, in 1959, Zipursky et al. Found that intact fetal cells are present in maternal plasma. Then it was shown that this approach might have implications for prenatal diagnostics. However, the main limitation of method is a low concentration of those intact fetal cells in maternal circulation. And the real breakthrough happened in 1997, when the detection of cell-free fetal DNA was discovered by Law et colleagues, and it basically launched a new era of non-invasive prenatal testing. Law then demonstrated that cell-free fetal DNA in the maternal plasma was detected by basically identifying Y chromosome fractions, and it was also used for RHD typing, and basically this was absolutely new field of diagnostics in clinical care of mothers. It was shown that the significant fraction of this DNA is released into the maternal circulation during basically the apoptosis of trophoblast, so it's not directly from the embryo, but from its trophoblast, and it means that DNA isolated from circulating fetal cells is actually placental. And the logical outcome of this research was the development of non-invasive prenatal testing. Non-invasive prenatal testing is basically screening for genetic diseases by using mostly the whole genome massively parallel sequencing of cell-free fetal DNA, which is in maternal plasma. We are able to identify this way basically not only sex status of the fetus, but also RHD typing, chromosomal aberrations, of course aneuploidies, and even other smaller chromosome microdeletions. This may be important to state that we are here using plasma to analyze DNA, which is maybe controversial, which was controversial by the time it was developed, because the plasma was discarded as the very first thing when we analyzed DNA, because it was thought to be the obstacle. So it may be a great lecture for the future scientists, that you can basically make interesting discoveries in the currently condemned fluids or tissues. Maybe the most important part or goal in development of non-invasive prenatal testing was the ability of the test to identify aneuploidies such as Down syndrome, Edwards syndrome, Patau syndrome. So the first and logical, most important step was to develop so-called trisomy test, which was also developed by the Slovak scientists. Most of them are currently working for Comenius University. It was a great success. And this trisomy test also identifies the three most common aneuploidies, which is, as mentioned, Down syndrome, Edwards syndrome, and Patau syndrome. And of course, with the advancement of the technology, it was able also to identify sex chromosome aneuploidies. From those, the most important are certainly a Turner syndrome, or so-called X-monosomy, and Klinefelter syndrome. And Klinefelter syndrome is basically caused by addition of X chromosome. And as we know, we have an X chromosome inactivation system in our bodies, and it basically prevents females from having additional genetic information, and most of the X chromosomes are inactivated. And while the majority of additional X chromosomes are actually inactivated, there is also so-called pseudo-autosomal region where the genes remain active even in those inactivated chromosomes. And this is the problem, because in Klinefelter syndrome, it is this additional genetic information that is still active that results, when multiplied by this syndrome, in those medical clinical problems we know from the clinical practice. Of course, via invasive prenatal testing, we can also look into the microdeletion syndromes. Maybe the most important are the George syndrome, which is caused by deletion of roughly 3 million base pairs region in the chromosome 22. Has frequency of 1 in 4,000 infants and causes congenital heart problems, specific facial features, developmental delays, and of course also learning problems, and very often we can see also a cleft palate. And of course, very famous is also Cret-du-Chat syndrome, which is translated from French as a cry of a cat, because the infants who have this disease have a very high-pitched cry similar to a cat, and it is basically a genetic disease that is in 90% of cases appearing de novo, so randomly. And it is also called 5p deletions, because you can see there that there is a short part of fifth chromosome almost entirely missing. It is important to realize that non-invasive prenatal testing has opened a new era of prenatal care, especially when it's coupled with advancements of bioinformatics, parallel DNA sequencing, automated pipelines, and all over together we are able to introduce this technology into the market and make it available actually for mothers all over the world to undergo. So I will just very quickly go through the workflow of this testing. It includes basically DNA sampling, which is from blood, of course. 10 millimeters of maternal blood is collected and then stabilized with EDTA. It is very important not to use heparin because it interferes with PCR reaction. Subsequently, self-free fetal DNA is extracted and it is selected, mainly the fragments which are about and shorter than 150 base pairs. And from those fragments, we are creating libraries which are then used for massively parallel sequencing. And after sequencing has happened, we are going to do a data analysis, which is of course nowadays automated. After years of scrupulous works of bioinformaticians became practically seamless to do. Now we can seamlessly go into another venture based also on non-invasive prenatal testing, because it is a well-known fact that cell-free nucleic acids are present in biological fluids independent of cells, and their increased presence of those cell-free DNA fragments in the blood was also proven in cancer patients, and it was in 1977, so it's not a newly discovered phenomena. And more so, it was observed that mutated KRAS gene sequence was detectable in plasma of cell-free DNA derived from pancreatic cancer patients, and this particular KRAS mutation was then found in tumor tissue of actual patient. And since then, mutations identified in cell-free DNA have been considered highly specific tumor biomarkers for diagnostics, determination of response to treatment, and of course the prediction of prognosis of the disease. And now let's just look briefly into the pros and cons of liquid biopsy. Because due to asymptomatic onset of cancer, most of the oncological patients are identified at advanced stage of disease, and too many times only by random diagnosis. The conventional tissue genotyping of cancer is standardized, but it is rather invasive, also very time-consuming, uncomfortable, and is not appropriate for periodic monitoring of the disease. On the other hand, the liquid biopsy offers a minimal invasiveness, very easy to obtain, quick. It offers a complex tissue profile, so you can see the human body actually from higher perspective. In the future, with the advancement of technology, it is also very great for real-time monitoring of the cancer patient. However, this approach is still not validated in clinical practice. In blood sampling, there are wide variety of technical and methodological aspects that need to be considered, and actually to avoid blood cell lysis and also genomic DNA contamination, it's still a big problem. Nonetheless, the prospects of this way of diagnostics is very promising, and there is an increasing evidence that shows that various types of liquid biopsy utilized for cancer-relevant biomarkers in patients, both male and female, can be used in future. Not only for diagnostics, but also for stratification, monitoring, and response to the therapy. And hopefully one day we'll remove absolutely from clinical practice often painful and dangerous and non-specific tissue biopsy. Just to briefly summarize, that what are we actually looking for in those methods of liquid biopsy in cancer diagnostics? It is certainly not only circulating tumor DNA, but those are also, for example, extracellular vesicles, circulating tumor cells, which are then designated for single-cell sequencing. Also, it's very rapidly developing field of microRNA, and of course, proteomics is also standardized approach for diagnosing of cancer or other diseases. And to sum up, what are the prospects and hopes we are putting into the liquid biopsy in cancer diagnostics? First and foremost, early detection is the holy grail, then monitoring of the disease progression, prediction to drug response. And of course, it is one of the most important tools for implying personalized medicine into the cancer care. And that catapults us into the last part of our today's lecture, and this personalized medicine. It is very important for you as medical students to understand and not to ignore this concept of medicine, because it is not just some kind of theory, but it's actually being implemented widely into some of the fields which we will mention. But in the future, really, it would be perhaps the only way for all fields of medical practice. So what actually personalized medicine is? It is based on fact that different patients with the same diagnosis might react differently on a particular treatment. Whereas a certain drug or therapy can have a positive effect on one patient, the very same treatment can be non-efficient or even potentially harmful or deadly for another patient. And the concept of personalized medicine considers mainly, but not exclusively, the genetic makeup of the patient. And what is another great feature and also benefit for the people and patients is that it shifts the emphasis in medicine from reaction into the prevention. But how did we get here into the personalized medicine? Well, let's look at the history briefly. First of all, for 100 years we had so-called intuitive medicine, which basically was very personal. We had the personal doctors, and each patient was really cared of like individuality. People knew some family history of the disease, and there were consultations, but physicians really couldn't do anything because they had no proven, or at least scientifically proven, drugs or treatments, so they just used their best guess, some herbal extracts, and that was it. Then, with advancement of science and with development of clinical trials, proper double-blinded clinical trials, we started to have evidence-based medicine, so we knew which drugs are efficient or non-efficient for a particular disease. But we treated patients as a population, not as individuals, and we didn't consider that they have a huge genotypic and phenotypic and environmental variability in them. So statistically it was better, certainly, but really the best would be, if you see those two pictures, would be to merge those two and create really personalized healthcare with science-based data. And we really need to understand this is a part of a complex system, and personalized medicine is actually disrupting paradigms of current healthcare systems. So there is a lot to be done and a lot to process. And for those of you who will not remain only in clinical practice but will do also research, we will see that this is a part of a bigger movement of actually ending of reductionism as a tool used to explain medical and biological phenomena, because we really see that there is no way to explain this by just looking into some physical data or biochemical data or genomic data. We really need to integrate those data into the complex scheme, and from that we have to deduce the ideal treatment for the patient. There are many fields, most of them relatively recently established, that are connected directly to personalized medicine. And it is important for you as medical students to have a really clear idea about what do those terms mean. So we can go right ahead and discuss, for example, genetics and genomics. Genetics is basically a study of genes as elements of heritability and their passing to generations. So we can associate genetics with Mendelian inheritance. Of course, what comes to mind first is classical genetic counseling for diseases such as hemophilia and others we saw today. And genomics, again, it's an interdisciplinary field of studying genome. It is complexity. So it is more focused on genome as a physical molecules, and it studies its behavior, it studies the mutations, its interactions, its dynamics, for example, gene therapy, and so on. So this is really a field that is undergoing the biggest revolution right now. Another two terms are pharmacogenetics and pharmacogenomics. Whereas pharmacogenetics refers to how variation in one single gene influences the response to a single drug, pharmacogenomics is a much broader term, and it studies how all genes of the genome influences overall response to a particular drug or treatment. So again, it is much more complex. Another two very popular terms nowadays are nutrigenetics and nutrigenomics. And whereas nutrigenetics provides information about how your specific genome should inform what you have to eat to maximize health. So for example, if you have a mutation that causes you cannot digest properly dairy products, you shouldn't eat dairy, simple as that, or to supplement lactase as an enzyme. But nutrigenomics focuses on how the nutrition you choose changes the gene expression, and these changes can have eventually big impact on your health and other stuff. So as an example, we can talk for example about resveratrol, and there were a lot of talks about so-called French paradox, that people who drink red wine have better health because resveratrol has direct impact on methylation status of your DNA, and actually where stimulating expression of proto-oncogenes protects people from the cancer, which is still very controversial today, by the way. But this is a good example of nutrigenomics, how the food you eat affects your genes. Now let's briefly discuss the tools that are used in personalized medicine. As we discussed previously, personalized medicine focuses on genes and our genetic makeup as a primary source of data to tailor the treatments, but we must extract those data somehow, and it is genotyping and sequencing that are most efficient and powerful tools. So when it comes to sequencing, I would not go into details since it was the part of your lectures, but here you have some interesting links for Illumina sequencing, which is the standard nowadays, and of course third generation sequencing, which is also called nanopore sequencing, where the main difference lies in that there is one particular sequence of a genome sequenced directly without PCR amplification of fragments and creating those libraries. And this has one major advantage: it is faster, it is cheaper, and there is no PCR bias, so there are no mistakes that were introduced via PCR amplification. I really can recommend those videos, they are very nice. But let's focus on genotyping right now. So what actually is a genotyping? Officially, it's a process of determining differences in genetic makeup of an individual by examining the individual's DNA sequence by using biological assays, and basically to comparing one individual sequence to another. In human genomics, genotyping also works in forms of assays when we design so-called assays, which contains mostly about 700,000 up to 1 million or even more positions in the genome, so-called SNPs, which are connected or associated with particular clinical relevance. So basically, instead of sequencing a whole genome, which is more than 3 billion base pairs, we are sequencing up to 1 million, but very relevant and very scrupulously selected sites, which is very efficient. And we are in real world able to obtain data about relevant part of patient's genetic makeup for about 100 euros. The process itself is a kind of mix-up of sequencing and microarray, which are methods you know already from the lectures. And of course, it is good to know that those are commercially available and successful so-called consumer's genomic kits. If you, for example, know 23andMe or DNA and so on, it is all based on SNP genotyping. Another very interesting venture of personalized medicine is the sequencing of human microbiome. Currently dominates certainly the intestinal microbiome analysis, so-called alpha diversity. What is important is that alpha diversity means in metagenomics that we are analyzing one site, basically all the organisms within the one site. For example, what would be the beta diversity would be if we would scan several places of human body and then compare the diversity. So this is basically all metagenomics, and it is performed methodically either by sequencing the whole DNA within the sample, which is very difficult for data processing. It identifies also viruses, phages, eukaryotes, but it is very helpful as well, and has potential to identify really every pathogen that we currently know and that was sequenced by us. On the other hand, there is, again, as with the whole genome sequencing and genotyping, this is the parallel, because we have a much more available and I would say economical method, and it is amplicon sequencing of so-called bacterial fingerprints, which are in 99% cases PCR products of 16S rRNA genes. Those genes, particularly their region 4, is ideal target for phylogenetic analysis, so we are able to distinguish species of microorganism based on this one region. So thus the nickname fingerprint. And of course, the outcome of this human gut microbiome analysis is the report, which you can see here, the examples. Those are usually about 40-50 pages long sheets, which are commercially available for, let's say, about 200 euros or 400 with consultations. And this report, which we see here, for example, is on the left focused on butyrate production pathways. Butyrate is very important short fatty chain acid, which is beneficial because it lining to insulin sensitivity and also makes us feel full, so it influences our satiety. And if we do not have enough bacteria which are producing butyrate, we need to either supplement butyrate, or we need to enrich our microbial microflora to promote the growth of bacteria which are actually fermenting cellulose and producing butyrate. On the right, we can see more advanced and more complex features of such examination. For example, you can see the so-called body axis. We all know that human microbiome is directly linked to our brain, heart, liver, skin. So we are able nowadays to check whether those axes are okay, and we can undergo some measures how to improve them. But of course, there is a lot to be studied again in this field, so there are about to be more and more scientific breakthroughs in this field. So when it comes to microbiome, and not only the gut but also the skin, mouth, ears, and many others, I really recommend you to watch this field closely, because it's very interesting, and there are going to be a lot of innovative discoveries. So in the end, the relevant question would be how to prepare for the age of personalized medicine. Well, this age already started in oncology, where there is really a horizon where we can see from 2016 to 2026 there will be 3 times more patients in the world that will receive the personalized healthcare, and pharma is not stagnating in this field as well. 72% of new drugs in development or in clinical trials are already designed for personalized healthcare. So oncology is really the pioneer of the personalized medicine for obvious reasons, because personalized medicine by the end of the day can save a lot of money, not only cost. And this is very important. I really cannot stress this enough. And when done properly and after initial investment into research and diagnostics, we are able really to save money and save lives and save comfort both to patients and healthcare system. Another issues are also education of patients, that they are able to obtain this kind of treatment. We already in Slovakia have some portals, for example, oncoinfo.sk, which was based by prominent Slovak researchers and doctors. Then again, there is a restructuring of healthcare system, but this is on another longer debate. But what certainly we can do here is the education of physicians and medical staff. And really this is something that applies directly to you, and it is very important for you not to ignore this topic and this trend, but to actively find the answers to the problematics of current medicine through the approach of personalized healthcare. As oncology already proven, this is the way to go, and other fields are only going to follow, so we have really exciting times ahead of us. And with that, I would like to conclude to this presentation. I would like to thank you also very much for your attention, and please do not hesitate to contact us if you want to know more about any of topics mentioned today. Here are credentials on me and my colleagues. You can contact us anytime and schedule a meeting or consultation via either telecom or phone. So I hope to see you live on the real lectures as soon as possible, and I'm wishing you a successful rest of the semester. Goodbye.